All right, so when we're looking at cladistics and finding the patterns of evolution, the patterns of inheritance in and among organisms, we have to think about what biology was from a historical perspective first. And when we do that, what we find is that biology, for most of biology, look at most of the time that modern biology has been around, it's been a descriptive science, meaning that the scientists that study biology, naturalists, what they would do is they would observe, draw, write, describe. They really wouldn't do much experimenting. When you look at what naturalists would normally have created, they created things like this: drawings of different animals in their natural habitat as evidence of their theories and of their relationships, and anything that was of a particular size or color or shape was named something, and that naming process was usually left to whoever discovered it or described it first, or had more political clout when the description occurred. And that's what most naturalists did, most biologists did in the beginning of the science. But when we look at, as time goes on, and we look at a real major step in the description of, in the development of biology as a science, we find Carl Linné coming along and defining things, giving a system to the nomenclature, and so giving a system to the naming of things, trying to show relationships and and describing their environments as well as the relationships between organisms. And so what Carl did is he defined kingdoms, families, genus, and species, what most of you are used to hearing in middle school. And he defined two kingdoms. He defined plants and animals. And that's all there was. And a lot of people, even today, if you took a census of the general population, they would be able to tell you, there's plants and then there's animals. If you pushed it and asked them about bacteria or algae, they would probably put all algae and plants, and all bacteria. They may call them an animal. There really isn't much more to the common experience than plant and animal, and that's not because of ignorance. That's because for a long time, and remember, we're talking hundreds of years, most people would look at it as something being either plant or as an animal, as a vegetable or as an animal. And what Carl Linné did is he defined it as that. He just said there's either it's a plant or an animal, and that's the kingdoms that we have, and then within those kingdoms there's families and there's genus and there's species. And he based it on Latin binomial systems, so that if you were given, for instance, Homo sapien, Homo would be the genus and sapien the species. So it's a wise man. However, when we look at the diversity of life, this system didn't work. As I just said, bacteria don't really fit into animal or plant, and a fungus doesn't really fit into animal or plant. If you look at the functioning of the organisms and how they do what they do, then this system really failed miserably. Darwin wrote his Descent with Modification in 1859. It was published. So not too long after Carl did his writing and his stuff was accepted. Even after Charles Darwin's ideas were accepted and all the evidence discussed and debated and then accepted by the scientific community, the system that Carl Linné developed did not really incorporate the evolutionary relationships between organisms. So if you had two mammals, let's say a duck-billed platypus was put in the same category as a grizzly bear or a human. And the differences in the relationships between these organisms were not described by their family categories. There was a whole bunch of different naming systems and categories that were developed, things like subspecies and etc. And even the word race for humans. Before 1969, really, that was it. They were trying to make a bad system fit. I'll use the word bad. It was the only system we had, but I'll use the word bad. In 1969, Whitaker defines a whole new kingdom. He says, you know what? Why don't we, not just a kingdom, he defined 5 kingdoms. He said, you know what? There's plants, there's animals, but there's also fungi, which are creatures that grow and eat undead things, but they don't do photosynthesis. He described protista, which are eukaryotic cells that single cell that swim around in fluids and eat bacteria and other microscopic organisms. And he described prokaryotes, so Monera, the kingdom of Monera, which was the bacteria. And it better explained the natural order. It incorporated the diversity better. It still didn't really take into account evolutionary relationships, but it did look at trying to address the missing diversity that exists in life. That actually exists. It just tried to describe that better. When we look at Carl Woese, and that's 69 to 77, so not too far, not too much longer. In 77, Carl Woese defines a whole new system with 3 domains. And in 77, he defined bacteria, archaea, and eukarya as 3 domains within which the other kingdoms could fall. So bacteria was given, was given its own domain. Archaea was given its own domain. So what we have is all the bacteria are here in on this branch of the phylogenetic tree, and this is the bacteria domain. In the domain of archaea, these are bacteria that are extremophiles. They are bacteria that do things differently, do different chemistry, live in really different environments, and accomplish things that bacteria cannot. And if we look then at eukarya, that kind of encompasses all of the rest of the life, right? So you have fungi here, you have plants here, animals, all the animals are over here on this branch, ciliates are over here, flagellates over here, and microsporidia over here. So these would be kind of the protista kingdom. There's the plant kingdom, there's a fungi kingdom, and animalia. So he kind of described this idea of having domains, and then all the other divisions of life could fall within these domains. It showed he did in this particular naming system really try to take into account the evolutionary relationships and the similarities and differences that each of the different organisms had. And specifically, he looked at a very conserved region of ribosomal RNA, and as you can see here, it's a 16S ribosomal RNA. And the technique he pioneered revolutionized the whole discipline of microbiology, because before then, you really, telling one species of bacteria from another species of bacteria was not an easy thing. And he actually looked at molecular biology. He looked at the small things you really couldn't tell to really describe the different species and in this case, his domains. Now, what we're talking about here is before this, it was either a round coccus bacteria, or it was a rod-shaped, or it had pili, or it had a flagella, or it didn't have a flagella. It was a spirochete. It kind of undulated through the liquid, or it didn't. That kind of stuff. When he came along, Woese came along before 77, but he published his stuff in 77. He said, you know what, we can use this new stuff that we have that we can look at, because before now we really couldn't look at it. but we have these techniques that we can actually take a look, use this gel electrophoresis. We can analyze little chunks of RNA, and we could really try to figure out what are the similarities between each of these and the relationships between the the molecular components that drive all the other big stuff. Okay, so then later, in around it really was between 1977 and 90 that we started to use in biology cladistics. And what cladistics does is takes everything into account. And this guy, Willy Hennig, he's a German, so I'm sure I'm pronouncing his name incorrectly. In 1950, so this is right soon after World War II, and I'm not even sure where he was working, if he was in the United States or if he was in Germany. He developed a system where he said, you know what? Why don't we try to organize things instead of trying to put them in these man-made characteristics, in these man-made groups? Why don't we try to look at what nature shows us and look at these particular traits in these particular and define whether these creatures have a trait or not. So there would be one group that stays outside of the clade. All right, so I'll walk you through it in just a second. So in this case, it would be the hagfish. The hagfish doesn't have a jaw, but before that, so this hagfish is kind of what we call the outgroup in this case. So this is the outgroup. Everyone else has a jaw. Now, as we move forward, so this whole thing's a clade, something with a jaw. All right, so this is the clade that's defined with things that have jaws. And as we move forward, the perch has a jaw, but you'll notice it doesn't have a lung. The perch came before lungs, because after this mark of time, every organism after that has a jaw. Then we could take a look at a salamander. A salamander doesn't have claws or nails. So everything after that has claws or nails. So this is the clade that has claws and nails, lungs and jaws. This is what this clade is. Notice we're not calling it a mammal or a bird or any specific stuff yet. We're just saying here among this group right here that we're going to define with this common ancestor. Notice this is a common ancestor. Please make a note that at each of these junctions, at each of these splits, is a common ancestor. Something that had claws, lungs, and jaws that gave rise to all four of these species. After the lizard formed, you'll note that at some point feathers formed. Everything that had claws, nails, lungs, jaws, and feathers, it came to a pigeon in this case. Of course, all birds could be on that particular branch. And if you develop firm mammary glands, then you're looking at a chimp or a mouse. So notice it says chimp or mouse here. And it's not mammals, because not all mammals have mammary glands. We know that because we look at a duck-billed platypus, and it does not have a mammary gland because it lays eggs. It doesn't even have a placenta, so that's one of the big things, one of the big divisions that we found later. But this allows us to take a look at the differences among what we used to group all in one group. So when we look at a duck-billed platypus, we don't have to put it all in one group now. We just look for a specific characteristic and see how nature presents itself. We're not imposing our own ideas on nature and trying to make it fit. So starting only in the 1990s, it might have started in the late 80s in laboratories, but I can tell you I didn't learn this till even after college. When I was in my master's classes, did we finally start to learn this in a more ubiquitous nature. Obviously, people were using this in laboratories, in specialty laboratories. But cladistics as a overall general topic to be taught in the classroom really was given root in the 90s, and it took off, and people are trying to use this now more often than ever. So let's take a look at how to make a cladogram and what exactly. That thing that we just looked at, by the way, is a cladogram. So when we're talking about creating it, and here's another example of it right here. This is a cladogram. What it is a line. That this arrow, this main line here, is time. Older organisms to the left and younger ones to the right. And what you see here are these characteristics, these traits. This is what we call an ancient trait. So over here would be something that didn't have that, and we don't have it here, but it would be something like, let's say, a squid. So a squid would be our outgroup, and that would be over here. So the squid would be our outgroup, and it sits out here. It does not have a vertebra. You could pick any organism, but it's just a comparison group. It's a control group, right? So here's our control group, our squid, our outgroup. An outgroup is a control group. And then everything else, this vertebrate developed, and then everything else is going to be put in our line. So if we're gonna put things in order, we need two ends, right? Our one end is the human, and the other end is the squid. The squid doesn't have any of these characteristics. They're outside. We're going to compare them, everything to this squid. So what we do when we have a group, we first choose the organisms that we want to try to group together. So again, the squid would be over here somewhere, and it wouldn't have any of these. It would be 0 to any of these. It wouldn't have any checks. So this is, each one of these has one of the characteristics we're looking at. So the key to doing this is to make some kind of grid, and what scientists do today is, instead of saying an X for yes and a blank for no, they'd say 1 for yes and 0 for no. And then they would use a computer program, which we will use in class, I hope. I'm going to try to make it work. I've signed out the Chromebooks for the next week or two, so we should be able to make it work on a computer program to allow you to do a statistical analysis once you get to understand what this whole grid is about and how we make this cladogram. Will allow you to use the statistical software to have the computer develop these most likely order of organisms in an evolutionary and the evolutionary relationship between them. So, and I want to emphasize that this is really common sense, right? So if we have 4 organisms: shark, bullfrog, kangaroo, and human, and we have 4 characteristics: vertebra, 2 pairs of limbs, which means 4, right? 4 limbs, 2 pairs, mammary glands, and then placenta. Then we have to go down the list and see if we can group them into groups that make sense. So let's go ahead and do that. So as a shark has a vertebra, but notice it doesn't have 2 pairs of limbs, and it doesn't have mammary glands, and it doesn't have placenta. So all those are left. So that would be on the outside. Now, if you're going to add your out group, it would be the squid. The squid would be out here somewhere. It's outside your whole clade. So when you're looking at the squid, it doesn't have any of these characteristics, but the first one to have the characteristic among these 4 is the shark. Everything within the outer circle, notice that everything within the outer circle has a vertebra. So the shark, the bullfrog, the kangaroo, and the human have a vertebra. Now the next thing to look at is which 2 characters, which organism has 2. We saw that which organism has 1, which organism has 2 characters. Well, we have bullfrog. A bullfrog is the next one that has 2. And since it has 2, it has vertebra. It's within the vertebra circle. But then we have to draw another circle to separate it from shark and kangaroo, because it has 2 pairs of limbs, but it doesn't have mammary glands or placenta. So here we have the bullfrog is all by itself inside its circle. And we move forward. We look for the next one, which has 3, 1, 2, 3, has vertebra, 2 limbs, and mammary glands. And that is inside the 3rd circle. Remembering that every circle inside has everything that the outer circle organisms have, but they have 1 or more characteristics that the outer circle organisms don't have. Or we don't call them organisms in this case, we'll call them taxa. What the taxa is the group of organisms that share a specific, usually a species, then we're looking at. So kangaroo is a taxa, bullfrog is a taxa, and shark is a taxa. When we look at clades, we're looking at defining a group of taxa that have a trait. And when we look at the cladogram down here, we'll discuss what are possible clades. So here we see kangaroo, of course, in this circle has 2 pairs of limbs, has vertebra, but also has mammary glands, but it doesn't have a placenta. And then we can see humans has everything every other organism has, and it also has a placenta. Now, I ask you this. If we accept, now remember, we're not talking about belief here or anything, we're using the theory of evolution. If we accept that evolution is a change, slow changes over time, which of these organisms, the one that has all the traits or the one that only has one of the traits, which is the oldest? Which came first? Well, we know the squid came first all the way over here because it doesn't have any of these structures, or at least it's outside it. At least it didn't. If a squid didn't, if we agree that all life on Earth came from an original source, then we have to agree that the squid is an ancestor at some point, but it certainly isn't an ancestor within this group, right? They're not close ancestor anyways. But which came first, the shark or the human? If we're all related and things change slowly over time. Well, we know the vertebrate. First of all, the vertebra came first because sharks have vertebra. But then 2 pairs of limbs. The bullfrog had it, but not the shark, so that must have come next. The shark only has 1 pair, and the bullfrog has 2. So the bullfrog must have came next. Then mammary glands developed at some point with some common ancestor here that did not include the bullfrog. The bullfrog is existing, it's hopping around, no problem. But some common ancestor developed. It's dead. This one's dead, and this common ancestor is here. And it developed this mammary gland, and the changes kept happening. Organisms survived or they didn't. And you get this branch we call kangaroos. Now, they're mammals that don't have placenta, but they do have mammary glands. So we call this organism, this taxa, kangaroo. And then we have placenta that came along, a development of a placenta, which ultimately humans are one of those mammals that have placenta. They give live birth. So there you have it. That's your cladogram. That's your that is how we make a cladogram. That is this is a phylogeny. It is an organization of taxa that tells you evolutionary relationships. Right? It's a tree. It's a branch of the tree of life that allows you to take an ancestral trait. In this case, we're looking at vertebra, and we pick an outgroup here. Right? We pick a group that's outside. It does not have that ancestral trait, and then once we get that ancestral trait, we start to see what the order of what the most likely, obviously it's most likely order of these taxa are in throughout the development of the various species. Okay, so let's take a look and actually do one now that we looked at this example. So here we have a grid. It has cells with flagella, symmetry, and bilateral symmetry. So symmetry at all you have here. Bilateral symmetry is something is they have symmetry on 2 sides. Mesoderm, so that's a layer of the tissue. Heads develop first, anus develops first, segmented body, calcified shell. Chitinous exoskeleton, water vascular system, and vertebrate. So we'll have to organize these in circles that make sense. So our first circle, let's go ahead and make a big circle. I'm going to try to do this here. Okay, so let's try to draw a big circle, and it's not going to be perfectly round. But there's your first circle, and the first circle is going to have all the organisms in it. So you'll notice here that All the organisms have cells with flagella. All of these create cells with flagella at some point in their development, including humans. Remember, sperm. So when we take a look at this drawing, obviously the one that has all of these characteristics, all of them, is the sponge, right? Oh, I'm sorry, I got it backwards. The one organism that only has the most rudimentary or ancestral trait of cells with flagella is the sponge. So the sponge is our outgroup. That's that organism that doesn't have any of the characteristics that follow. It only has one ancient characteristic that we're going to call that we have decided was important, and that was cells with flagella. So a sponge has cells with flagella, but doesn't have any of the other characteristics that would allow it to be in the circle. A clade is in the circle. Anything that's a clade is within the circle. And a sponge is outside the circle. Now the next organism that has symmetry but none of the other characteristics, so it has symmetry but doesn't have any of the other characteristics, is jellyfish. So our next layer of our cladogram, so organisms that are within the circle, the black circle, have one set of characteristics. The jellyfish, and let's go ahead and put it here, jellyfish have cells of flagella and they have symmetry, but they don't have any of the other characteristics. Okay. So now let's take a look at the next layer. Which organism would be next in our discussion, in our description of what kind of circles we could possibly make that would make sense. So the next circle has to be flatworm. You'll notice the flatworm has cells of flagella, so that's within the blue circle. All of them have it. It has symmetry, so it's within the black circle. Now the question is, does it have bilateral symmetry? It does. So it would be the next layer. In the next layer you have bilateral symmetry. And so we're looking at then the flatworm. So the flatworm is inside that circle. So the flatworm has cells of flagella, because that's everything on the diagram. It has symmetry, that's the jellyfish, and everything within it has symmetry. And then the flatworm has bilateral symmetry, so everything within there also has bilateral symmetry. The next thing that we see that's on our chart here is mesoderm. So which of these now that's something the flatworm doesn't have, but everything else on the chart does. So we'd have to draw another. Well, that's everything within this line here. Everything within this second blue line has a mesoderm, right? That's everything. So the flatworm doesn't. That's why it's not within this circle. Again, clade. Each circle defines its own clade, right? All right. So the next thing is heads develop first. Well, starfish and humans, their heads don't develop first. Their anus develops first. Brings a whole new meaning to butthead, I guess. But anyways, anus is developed first with humans and starfish. But with earthworms, snails, and fruit flies, the head develops first. So how would we be able to define a group that has heads develop first and anus develops first? So the way we would do this is we would make two circles, right? So within the idea of everything in this circle has mesoderm, that's according to this chart, right? All the organisms have mesoderm. They're all within this circle. Heads develop first, only these three are there. So we draw one big circle here. here in this big circle, these organisms all have heads develop first, and the organisms on the other side, another circle, all the organisms in this circle have anus develops first. Now, if you take a look at this, that means that this group here, this is one group, and then this is another group, right? Anus develops first here, right? Heads develops first here. We take a look at these two, there's a difference. One has a water vascular system, the other one's a vertebrate. So there's two circles within this circle, right? So we would go ahead and draw a circle in here that looks at, and what's within the only organism that's within this circle, which would be, let's say, water vascular system, would be the starfish. And everything in this other circle next to it, and let's go ahead and do it in red just to show a difference, is going to be vertebrate, anything with a vertebrate, and that's humans. Now, in this bigger circle over here, we have three organisms. Two of them share a characteristic, the third doesn't. So earthworms and fruit flies share a segmented body. So they are within one circle. They have, in this circle, you're going to have both earthworms and fruit flies. But you'll notice that fruit flies have a chitinous exoskeleton. Chitin is a protein that forms the outer shell of a lot of insects. So fruit flies have a chitinous exoskeleton, while the earthworm doesn't, even though they both have segmented bodies. So this particular, the inside a third circle, another circle, you're going to have fruit fly. And the fruit fly is inside the circle, in the red circle, because it has a segmented body, but it has a chitinous exoskeleton, where the earthworm, notice here, the earthworm does not have a chitinous exoskeleton, so the earthworm doesn't get that, doesn't get to be within this circle here rather. So the earthworm needs to be out here. Now we look at where we're going to put, what was the organism that would go here on the outside of this fruit fly, right? So within this circle of a segmented body, of head develops first, we're looking at the segmented body that would be both the earthworm and the fruit fly. Outside, right? Outside of that would be this calcified shell, would be the snail. So the snail has to be in a circle all by itself that has snail in it, because the snail has this calcified shell where the other two don't. But both these creatures, the snail and the earthworm and the fruit fly, they all have a head develops first, which is this circle. All of these organisms here, starfish and humans, they both have anus develops first, where starfish has a water vascular system, where humans have a vertebra. All right, so that organized us, but now we have to draw our phylogenetic tree, our tree of life, our family tree for these organisms. It's a family tree. You've all drawn family trees in middle school, I'm sure, or have seen them at least, and I think it's clear that this helps organize a relationship based on characteristics, based on something you can observe and statistically analyze. So let's go ahead and draw a line. We did our Venn diagram here. Let's draw our cladogram. So this is how things developed over time. There's our time. The very first thing that's over here is a sponge. And what developed with the sponge was this thing. This thing had cells with flagella, right? And cells with flagella. Then what came next was, so here's cells with flagella, right? Before that, but the first thing that came along that organized everything that came after it was this idea of symmetry. And after symmetry came the jellyfish. So this branch came off of here, some common ancestor that formed a jellyfish. And you see that the jellyfish is out here. It's in the outermost circle, and it's our first organism on our clade, on our group. This being the outgroup, right? Then bilateral symmetry came along as a characteristic. Some kind of evolutionary event happened, some mutation, and you had bilateral symmetry, and that was very successful. Bilateral symmetry started to develop all kinds of organisms, and you can see them here inside the circle, inside this black circle, including flatworms. So flatworms were the next thing to come off right after you develop this thing called bilateral symmetry. All right, and as you had your bilateral symmetry, the next organism to come off of the tree here was this flatworm. Okay, so the next thing that developed over time, bilateral symmetry happened. You got the flatworm, and everything that came after that on the line has bilateral symmetry. Then soon after that came this idea of mesoderm, this trait of mesoderm. So after the flatworm, all the other organisms had mesoderm. Everything before it didn't. So that was the next development right here, mesoderm. And as the mesoderm developed, that allowed for other things to occur, and those other things were the mesoderm. Again, this is the line for the mesoderm, right after the flatworm. So all these organisms still have to come along, but mesoderm had, everything has a mesoderm, but head developed first. So two things happen. You had the head developed first group, and then you had something else happen. So something came along and head developed first. So we had a branch. All right, that way, and I guess I shouldn't put an arrowhead on it. Shouldn't even make it that long, I guess. And off this branch, of course, here was head first, right? So the head developed first there, and of course that branched off into two groups, right? One group kept going in the other group, and the head developed first group. Oh, there are three, I'm sorry. So two groups, these two had something in common, but this one branched off all by itself. That was head developed first and calcified shell. So this is the calcified shell. Now, you had the calcified shell. Of course, at the end of that, you're going to have snail. Over here, on the other branch, it branched off twice again. Here's head developed first. It doesn't have a calcified shell, but it does head develop first. That's correct. It doesn't have calcified shell. That's why the snail's off by itself. No, the snail's off by itself. The next two things is earthworm and fruit fly. Earthworm has a segment. They both have segmented bodies. That's why that's what this was here. Bodies. And they both have that. But where one, after segmented bodies, these two have segmented bodies, the fruit fly has a chitinous skeleton. So at the end, the worm, the earthworm, is here, where the fruit fly is here, and of course the thing that the fruit fly has that the earthworm didn't. If you look at the chart, the fruit fly had a chitinous skeleton. So I'm just going to use blue and say CHSK for chitinous skeleton. So what we see here is a symmetry. Started with symmetry. First you had a jellyfish, then it developed a bilateral symmetry that gave you everything that came after it, and then the flatworm came along. But the common ancestors that were not part of the flatworm group, they developed something called mesoderm, which gave them some kind of advantage. And as they went down the line, some of them started developing head first, which then allowed the snail to form, because the snail developed a calcified shell. But the other group from this common ancestor formed earthworms and fruit flies, because earthworms, they have segmented bodies, but they don't have the chitinous exoskeleton. So the fruit fly, which is another part of the insect group, that branched off this common ancestor. Now, as we move forward, we still have two more groups to put here. What we find is that we can, and it's a little messy, I know, but I hope you get the idea, that as we move forward in time, we still have, again, something that has mesoderm, does not develop first. In fact, has what? Anus develops first. And because the anus develops first, these two groups end up being together. You can see them here, the starfish and the human, where the starfish has water vascular system, so we branch off soon after that, and you get the starfish, and that's because the water vascular system. And of course at the end you have the human, and that's because the human has the vertebra. And that's kind of a messy cladogram, but it is in fact a tree, right? We have a sponge that's outside the group, then something called symmetry happened, then this, of course, the sponge has something the rest of them do as well, that's cells with flagella. So this has an ancient characteristic, where here we developed symmetry a little more recent in time, and jellyfish came off. They have symmetry, they have cells with flagella, but they don't have bilateral symmetry. Everything that came after that had bilateral symmetry. And here you have this branch of the flatworm, because the flatworm had bilateral symmetry but not mesoderm. And as you take a look, you know, so here the mesoderm developed, and then all of a sudden you had some common ancestors. It had mesoderm, it had bilateral symmetry, had symmetry, it had bilateral symmetry and mesoderm, and so that's your common ancestor. But here head started to develop first. That led to snail, because the snail developed a calcified shell, so that led you to a snail. And then some other organism, somewhere along the line, some evolution, some mutation allowed for segmented bodies, and then that allowed for the earthworm to be where it is, and which has a segmented bodies. Head develops first. It has mesoderm and bilateral symmetry and symmetry in general. And here, a chitinous skeleton mutation allowed for a fruit fly to come along, because it has a segmented body, and it has a chitinous skeleton. And here on this branch, from that common ancestor that didn't have either one of these, right, or had both, or whatever. Here's anus developed first, right? And from the anus developing first, the starfish came along because it had a water vascular system, so it allowed for the starfish to develop into this tax of starfish came off of this water vascular system. Well, this common ancestor didn't have it. This one, as soon as that mutation happened, you got this taxa here called starfish. Those ancestors kept developing and living and dying and whatever else happened. And then all of a sudden you had vertebra develop. The vertebra developed, some mutation allowed vertebra to happen. And as soon as that happened, some time after that, you have humans. So in the time scale, this tells you how the order of their development and the relationship. You can then start to organize different taxa. So you can say, well, everything in here is a taxa. I mean, not a taxa, excuse me, a clade. Everything with this common ancestor right here, that's a clade. We're going to call it the bilateral symmetry, or the symmetry clade. Everything in there has symmetry. And we can say everything in in here, we'll call this a clade as well. And we'll say that everything in here has mesoderm. So everything with mesoderm is, this is the mesoderm clade. This is the symmetry clade, and then this is the mesoderm clade. And you can do this all day long. You can decide what clades you want to look at, and you want to compare different relationships and different evolutionary events, and you can define these clades all day long. It's called tree thinking. It's called cladistics, and you're talking about producing a phylogenetic tree. All right, so let's go on and let's take a look at what you were asked to read. This I'm not going to go and read it to you. I sent you an annotated copy where I wrote down some notes for you on the side and highlighted some important parts. It talks about exactly what I've said already as far as the history of classification and its importance, and the idea that we don't want to be imposing our own organization to how animals are related to one another, but we want to discover what nature has given us. Here again, I've gone over this part of the reading in the previous section. You should look at the annotated version and make sure you understand, and of course the timeline that I created, and make sure that you look at the difference between the Linnaean model, the 5 kingdom model, and Woolsey's system that looked at 3 domains, and notice the difference in how each model was able to better describe the organization of life as we find it. In the first model, that was Linnaean's model, we found that it was lacking in its diversity. In the second model, it increased its ability to describe the diversity in nature, but still lacked the description of the tiniest of life, specifically bacteria, and the diversity found there. And Woolsey was able to better describe that diversity by creating these 3 domains. Of course, all of these models still lacked the depth of trying to classify life by the evolutionary realities, the evolutionary relationships between the organisms. Carl Woolsey's organization was probably the best so far, and why it's been accepted thus far. He based his on molecular information, specifically ribosomal RNA, and the comparison there that allowed him to describe a domain, a difference between bacteria. But then we moved into cladistics and trying to analyze taxons. Remember, taxons are what you might call a group of organisms, who you might call a species, right? So try to describe taxons in terms of how and when they developed specific character traits. And the creation, the result of that organization is a phylogenetic tree. So that's again why we talk about tree thinking. Here you have Carl Woese's version of the phylogenetic tree, and here's the 5 kingdoms. Here you'll notice that Monera was all put into one kingdom, where here we're talking about 3 domains, and each domain then had a breaking it down into several parts. And if you look at Woese's 3 domains, you'll see that the tree has animals, fungi, plants, and protists, single-celled eukaryotic organisms, all of them being eukaryotes. The 2 prokaryotic or lacking nuclei branches of bacteria and archaea are significantly different in the way they process energy and the way they the macromolecules that make up what they are. So Woese's system takes into account molecular data and organizes the tree in again a better description of what actually is in nature. Now here is the process that I tried to describe to you earlier in the examples, where we looked at several different organisms and we created a tree. We created a cladogram. And these are cladograms, or phylogenetic trees. And I guess there's some key terms that you need to know. And again, you should have looked at the annotated version. You should have read it yourself, looked at the annotated version, and taken notes. But when we look at the taxa, all right, a taxa is a group of organisms. All right, so it could be humans, it could be cats, it can be birds, or whatever it is that you're looking at. It's general enough that it allows a scientist and allows you to decide what that taxa is going to be. It's not limiting the way the problem of the duck-billed platypus was, and the definition of mammals, and the issue they had trying to put oviparous mammals into non-placental mammals into the category of mammals, because mammals had a very specific definition and it was difficult to place them there. Well, here you have taxa, and so this is a much more general term. It allows you to be a little more flexible and yet be able to set your variables in a very clear and precise way. They're arranged into clades and sister taxa, which are taxas that are closely related, the most closely related ones. They fall into what's called a monophyletic tree. And what that means is that these organisms here, at the end of each of these lines is an organism. 1, 2, 3, 4, 5, 6, 7 organisms. They all share a characteristic. So it could be placenta. Maybe this was the characteristic placenta. You remember in the examples that I worked out early on, you had a character, and I put the character, a little line out for the character, and then everything after that had that particular trait. Well, in this case, this tree here, they share a common ancestor that had a particular trait. In this case, I'm saying it could be a placenta, which might mean monkey, ape, monkey, gibbon, chimpanzee, human. It could be cow, oxen, or what have you, and a particular trait that all of these organisms have placenta. So you couldn't have a kangaroo here because a kangaroo doesn't have placenta, and you wouldn't have a duck-billed platypus here because this doesn't have none of these have placenta. So a character, a monophyletic branch of a tree, or a cladogram, a clade. A clade is a group of organisms that share a specific character trait. So if you're looking at an organism where you have a common ancestor, so that defines the clade, they all share that derived characteristic. That's a monophyletic tree, or monophyletic branch. Where if you're looking at a group that's polyphyletic, they have a common ancestor, but not all of them, when you're looking at a group of organisms that share a trait, not everyone with that particular common ancestor has that trait. So here and here, for instance, these two don't have that trait. Maybe they lost it. Maybe there was an evolutionary. Maybe they had placenta. This ancestor had a placenta, and at some point, this particular beast and this particular piece, the possum in America, and over here, it could be the kangaroo in Australia, lost the placenta and developed another method of offspring, developing offspring. Maybe this is the duck-billed platypus and this is the possum, or something like that, where you had some common characteristic that all of the ancestors, all of the progeny, all the lineage of this particular ancestor have this trait, except for two. And again, it could be lost, or it could be a derived trait that 5 out of the 7 actually derived. We call that a paraphyletic tree. So you have some, but not all. Now, polyphyletic, you're looking at it. So in a polyphyletic tree, you have individuals in a group that have more than one ancestor, more than one common ancestor. So in this particular example, this is a common ancestor here, common ancestor, this is another common ancestor, here's another common ancestor. And this particular group would be called a polyphyletic tree. Now, it's not just specific characteristics, morphologies, shapes, and sizes that are important when you're creating a tree like this, as we saw what happened with Woolsey and the development of the 3 domains. You can actually look at DNA or RNA in the case of Woolsey and look for synaptomorphies. Those are shared derived characteristics and you can focus on the molecular characters. It doesn't have to be a morphological character. And synaptomorphies are when you're looking at shared derived characters, so characters or characteristics that are shared among the individuals in the groups in the particular clade that you're looking at, and they're derived. In other words, when you're looking at derived characters, you're looking at characters or characteristics that have evolved over time from some ancestor that developed the clade. So some, here again, to go back to the placenta example, this particular trait or characteristic was placenta, and all of the organisms under study, every member of this particular clade has that shared evolved, or another word for evolved, derived character. Now, there's an assumption here, and that's that that all these characters were formed from homology. In other words, they evolved from a common ancestor, right? So homology is this idea that this trait all came from this organism, a common organism here, a common taxa here, not organism but taxa, that each of these have inherited from that particular ancestor. We call that homology. But there are a couple of other ways to get, and there's several ways to get at to this end. There's and that would help explain these polyphyletic examples and paraphyletic examples. And that's this idea of convergence. That's where circumstances, natural selection, have made something so advantageous that particular trait evolved more than once, even though they did not evolve from the same common ancestor. So a great example of that is the one that's described later in the reading, and that's the flying. Flying developed in butterflies, in other insects. Flying developed in bats, and flying developed in birds. Butterflies, birds, and bats have different sets of common ancestors. Of course, if we go back far enough in a tree, we're all related to one another, but each of them evolved from groups that did not have flight. So bats evolved from mammals, and mammals did not have flight at some point in their ancestry. Birds are dinosaurs, and dinosaurs did not fly until they finally developed wings, or at least flight evolved at least twice, right? In the dinosaur lineage, as all of you, I'm sure, remember the pterodactyl, and then you have the development of flight via feathers and hollow bones and etc. that we call birds today. That is another lineage. So flight developed twice there, and of course, among insects, the development of flight at different developmental stages. So each of those found 3 different ways of developing that trait, that characteristic, flying. Now those would be a convergence, and that's where you get something that's so advantageous that it's able to that natural selection pushes organisms to develop that particular character state. Of course, you get reversals as well. So something could evolve these particular character states, let's say flying, and at some point you get a change, a more evolutionary pressure, and these organisms no longer have that derived trait. They've evolved actually backwards, right? So it's inaccurate to say backwards, because backwards implies that there's a direction, and there's no direction, right? So, but it is a reversal of, just means that something developed a particular character trait, and then moving forward in time, some long forward progeny loses that particular character for whatever reason and reverts back to an ancestral version, keeping many other of the character traits that they shared with the other taxa that developed from that lineage. So what would be an example of that would be a whale, right? A whale, the ancestors of whales crawled out of the ocean. Like we share that one common ancestor, that tetrapod, that fish that over time, over many small changes, developed the ability to breathe air, lungs. An example of that would be a lungfish, right? But this ancient ancestor of ours, they crawled out of the ocean and then led to all animals that walk the earth. That particular tetrapod, 4-legged, 2-paired organism, right, is on land. Later developed, and you had pigs, and then you had hippopotami. And so the hippopotamus and whale had a common ancestor. One group of those ancestors went off and kept hanging out near water. And over time, those organisms, that particular taxa, and started to change. And natural selection started to select those organisms that could spend more and more time in the water and go deeper in the water. And the legs eventually, they didn't turn into flippers. That's something a lot of people mistake in the concept of evolution. They didn't actually change. The evolution means that through very small changes over a long period of time, the appendages of the ancestors of whales turned into flippers, right, and a tail. And you can still see the bones of a rudimentary legs in a whale. You can see ankle bones in whales, in modern whales, and then ancient whales. And you can see those little bones that are still there. They're vestigial, which means they don't have any purpose in the whale. But their ancestors had those bones. But they've developed then the ability to swim and hold their breath over lengthy periods of time, but they lost the ability to walk on land. That ability to walk on land was a common derived trait that all mammals had, and this particular lineage lost that trait and redeveloped the flippers that fish have. And so you get whales and dolphins, all from that same group of taxa that evolved back and lost the ability to walk on land. Flight. Let's look at flying. Birds develop flying over many millions of years, but then you can look at a penguin who can no longer fly, or an ostrich that can no longer fly. They gave up that ability to fly in order to revert back to some other character state. Again, it wasn't an individual that lost it. It wasn't even a taxa that lost it. It's a very gradual changes over a very long period of time that led to the modern ostrich and the modern penguins. But penguins and ostriches have lost the ability to fly. And that was a character trait that took a long time to evolve or derive. So here is a fairly decent description of the parsimony analysis that we did in the example that I started this video with. And that is the idea that you're basing your logical decisions on Occam's razor. And Occam's razor is just simply the idea that the simplest theory is the most likely theory. So Occam was a monk who had studied logic and mathematics and had come up with some basic logical statements that would transform over time into this idea that to test something, if you have two competing theories that are exactly the same, have exactly the same outcome, but two different reasons for the outcome, the simplest answer is the best answer. So parsimony is based on that logical rule of thumb, and that's that if you have more than one tree, if you end up with more than one tree, it's the simplest answer that's the best answer. For instance, let's take a look at taxa 1 here. And there's these 1, 2, 3, 4, 5, 3, 4, 5, 6, 7, 8 different characteristics. Would it be more likely that this particular taxa was developed later in the evolution of organisms and therefore had all the characteristics of all its sister taxa, or taxa that developed along throughout the process? Or is it that this particular taxa 1 developed A, B, C, and D along with everyone else, then lost A, B, C, and D, developed E, F, G, H, and then redeveloped A, B, C, D? No, the more likely answer is that taxa 1 is an organism, or is a taxa that is further down the timeline and was the beneficiary of all these various evolutionary steps that led these particular characteristics. This particular taxa 4 did not have any of the characteristics except 1. So is it more likely that it had all the characteristics and then lost all of them except 1? Or is it that it's a earlier taxa that developed just one characteristic but never developed any of the others? It's probably more likely that it's 4, that it never developed the others, it just developed one, and that was a very successful evolutionary step for this particular taxa, and it never had to change, and it was never, natural selection never selected it out of the equation. So when you're looking at these 4 possible trees down here, rooted trees, any unrooted tree, any tree that looks like this without roots meaning there's no sense of direction of which one came first, second, or last, but rather just an idea of how closely a particular group is related to another. Once you decide on the order of these evolutionary steps, the question is which one is the simplest to get to your outcome, to your which one of these four theories explains the result in the simplest possible way. And in fact, sometimes you have more than one right answer, and it takes more time to more research and more facts to help you decide which of the four is the most likely. So in the application of molecular phylogenetics, you're looking at, they're trying to describe here how DNA and RNA can be used to try to define which one of those trees is the most likely, and in fact maybe even define some of those trees. And there's some really good, really solid positives to the idea of using molecular information. A lot, as we've discussed in class, a lot of the characters that make up an organism are unseen. You don't see the particular trait, and so looking at molecular data helps us see those. Then here they're talking about the unseen changes in nucleotides that are, in other words, are not impacted by natural selection. So what they want to say here, I think, is that when you're looking at evolution, there's a giant chunks of DNA that natural selection doesn't really impact, and they're only subject to mutation and to random genetic drift. And so because natural selection isn't having any kind of pressure on these particular gene sequences, only time actually impacts those nucleotide changes. So when you have two morphologically similar organisms, and you can actually start to see how much time elapsed between two species, one might be older than the other, because of these regions of genetic drift that they're able to look at. And again, you could take a look at Carl Woese's work earlier on as an example of being able to use something that has a very conservative region of DNA, or in the case RNA, that allowed that particular region to help Carl Woese identify which organism came first in the tree of life and which came later, and in fact define an entire domain of life. So here phylogenetics was actually used to identify, in this particular case, whether an individual gave his patients HIV. If the way they were able to do that is look at those regions and compare the changes that happen in that particular virus. And because HIV changes so quickly, it actually allowed them to have enough difference between the HIV strains to be able to identify that which ones were related to which, and they were able to bring it back and identify that the dentist indeed did infect his patients with HIV. Here we can look at the idea of the origin of bats in flight, and there's two alternate possibilities. All right, the phylogeny here, where you have a monophily, where you have one where flight developed one time between, which then led to microbats and megabats, and from some common ancestor that all us mammals have. There's some kind of placenta here that, some down here it might be the first organism with a placenta, let's say, and then there's all these different mammals came from that. Placental mammals came from that. Or you could have had this flight might have developed twice. Maybe, if you think about it, if you just looked at the morphology, just the shapes and sizes of the organisms, you can imagine someone saying that a primate is more closely related to a rodent than a bat is to a rodent would not fly well, forgive the pun, with a lot of people. Where if you said a microbat was more closely related to a rodent and the megabat was more closely related to primate, well, at least they have size in common. As it turns out, you know, when look at, and you could even apply Occam's razor to this, is it more likely that flight would have developed one time and allowed these two bats later, you know, the smaller version being a microbat and the larger the megabat? Or is it more likely that flight actually developed twice, that two organisms so similar to each other could have developed from some common ancestor and just developed wings, and they were just bigger here, and these are the bigger of the ancestors of this particular common ancestor, or rather the progeny of this particular common ancestor, and the smaller versions of the progeny of this ancestor, one of those developed flight again. So, you know, Occam's razor would say that it's the one that allowed us to develop the particular character with only one change is the more likely. And as it turns out, if you did the reading, it is actually this one that is the most likely evolutionary relationship between bats. In this particular region, they talk about some of the evidence, the molecular evidence between that allowed them to understand the evolutionary relationship between megabat and microbats. And it reminds us that AG switch more easily than, and CT switch more easily than A with T or G with C. And that these particular combinations kind of insinuated that there might have been a convergence happening. In other words, two separate lineages each developed flight independent of each other, and they argued that the regions of DNA may have suggested that such an event occurred. But once you looked at the most parsimonious phylogenies, and you look at the idea that these two developed from a common ancestor, because again, it's the most likely reality, considering that developing flight twice in two different lineages might seem a little difficult given a certain amount of time. Molecular data, once fully understood and defined, and statistical computer programs eventually showed that it was that these two bats came from the same lineage. Another example of evolutionary history where things, and we've discussed it already, is whales, and that particular example is on your video that you'll watch as well. You have to think about, though, when you're talking about using any kind of evidence, but genetic material specifically is difficult to adds to the difficulty of the use of genetic material, and that is the idea of lateral gene transfer. And what that means is that organisms that are not related to each other can receive gene sequences from other species, and especially bacteria. This tree, as discussed in the video, shows you these lateral gene transfers, especially early on in the development of life, which seems to have been the most where it happened most of the time. However, bacteria do this all the time, as you can find out from the reading, in several different ways, including conjugation. It's important to understand that higher forms of organisms do it a little less. You'll notice that bacteria do a lot more. Here they are transferring DNA from one from the other, where here's chloroplast being transferred over to eukarya, and mitochondria coming over here. Notice that mitochondria came in earlier than chloroplast did. You'll notice that these two events were the endosymbiosis that we had discussed earlier in the year. To the end of the video, in this particular video, it's been a long one. I'm going to upload it now, and I'll create the TED-Ed and then send it to you. I hope this helps you understand your document and the idea of cladograms.